dear viewers. So today we will be discussing nucleotide excision repair. So what actually is excision? Excision is the process where, suppose this is some strand or some long rope. If we cut it at 2 points and remove the in-between portion like this portion, and join these two ends here and here, we can say we have excised a part of this rope. So when we cut something from 2 points and remove the in-between part, then we can say we have excised this part out of this original rope or anything. So that process is called excision. So let's see how the nucleotides are excised from the DNA molecule. This is our DNA molecule. As we all know, it's made up of the two strands. Suppose this is one strand, this is the other strand. This is our thymine bases, thymine and thymine, which are bonded correctly with the adenine bases with two hydrogen bonds. Sometimes what happens, mostly in the case of pyrimidine bases, they get to form bond with each other. Now in cell of E. coli, this thymine dimer has been formed. So now E. coli cell has to remove this thymine dimer. It's done with the help of a UVRABC excinuclease complex. This complex actually is made up of many proteins like UVRA, UVRB, UVRC, UVRD, and like this many more. Ultimately, it's known as the UVRABC excinuclease complex. Why excinuclease? See, suppose this is the DNA molecule, and if any protein cuts it from one side and remove the nucleotide, suppose from this end or from this end, we know then we call these proteins as the exonucleases or the exonuclease enzymes. Suppose any protein cuts it from in between, then these are known as the endonucleases. Suppose any protein makes two nick in the DNA molecule and removes the in between portion, then such kind of proteins are known as the excinucleases. So ultimately this kind of repair is done with the help of excinuclease protein complex. So first of all, this defect has to be detected. It's done with the help of UVRA protein. This protein UVRA, it detects that there is some kind of defect in the DNA molecule, because when the dimer is formed, this portion it gets distorted. See, here a dimer is being formed. So now DNA chain, its configuration changes and this portion gets distorted like this. When this dimer is formed, the adjacent nitrogenous bases, their hydrogen bonds also gets broken because of the formation of this dimer and change in the configuration. So this change in the configuration gets detected with the help of UVRA protein. So after that, UVRB protein gets recruited here. This protein comes and gets bound to this distorted portion, this red portion. This protein is UVRB protein, and it results in bending of the DNA molecule. So DNA molecule gets bent like this. First the defect gets detected with UVRA protein, then UVRB protein gets recruited here, and this protein results in the bending of the DNA molecule. After this, UVRC protein comes. After this bending of the DNA molecule with UVRB, now what happens, UVRC protein, it comes here and it gets bound to the adjacent portions of the bent. See, this is the bent angle. See, this is the angle where the DNA molecule has been bent by UVRB. Now this UVRC protein comes and it gets bound to the adjacent portions of the DNA molecule. This is the main protein which forms two nicks in the DNA molecule. So where it forms the nicks? First nick is made around 8 bases upstream in the DNA molecule. So suppose this is the 5' end of the DNA molecule, this strand, this is the 3' end, and the opposite strand we know is always anti-parallel. So this is the 3' end and this is the 5' end of the other strand. UVRC protein forms two nicks. First is the around 8 nucleotides upstream in one strand, and the other nick is around 4 nucleotides downstream of the defect. This is our defect, this is the thymine, this portion is the thymine dimer and the defect lies here. So first nick is made 8 nucleotides upstream. This is the first nucleotide, second, third, fourth, fifth, sixth, seventh, eighth. So after the 8th nucleotide, at this portion, one nick is made and the other nick is made in the same strand around 4 nucleotides downstream. Downstream means towards the 3' end. So this is the defect here. This is first nucleotide, second, third and fourth. So the second nick is made 4 nucleotides downstream. So in this way, this portion is ready to be cut now. We can remove this portion because the ends have already been cleaved. Now this portion is only attached to the other strand with the help of the hydrogen bonds in between the nitrogenous bases. So now UVRD protein comes. This UVRD protein is actually a helicase. See, helicases are of two types. One is helicase 1, one class is of helicase 1. The other class contains the helicase 2 type of enzymes. So helicase 1 actually, they cause the unwinding of the DNA molecule or the unwinding of the DNA strands. And the helicase 2 class enzymes, they can do the unwinding also and they can perform many more functions also. So here in nucleotide excision repair, basically UVRD, which is a type of helicase 2, this enzymes comes. So this can cause the unwinding also and it can remove the nucleotides also. So here it comes and removes this portion, this whole portion in between the nicks. See, from here till here, in between the two nicks made by the UVRC protein. So ultimately this defected portion gets removed along with many nucleotides upstreams and some nucleotides downstream of the DNA strand. So we have the remaining portion like this now. This is the other strand which was right. This is a defected strand and this portion has been removed. The portion in between the two nicks made by the UVRC protein. So after this, DNA polymerase 1 comes and it attaches the nucleotides from 5' to 3' end like this. It will attach the correct nucleotides here and ultimately one nick is left here and that nick is ligated with the help of DNA ligase. So in this way we get to have the DNA molecule without any defect with the correct strands. So let's see the whole process once again. See, this is a DNA molecule. It had two thymine bases adjacent to each other. These thymine bases resulted in the formation of a bond in between the two and they formed a dimer. Now E. coli cell had to remove this dimer. It's done with the help of UVR-ABC excinuclease complex, which is a complex made up of many proteins. One of the proteins is UVR-A. It actually detects the defect. It actually detects the dimer here. Because wherever dimer is there, that portion gets distorted. So UVR-A detects the defect. After that, it recruits the UVR-B protein. UVR-B protein results in the bending of the DNA molecule where the defect lies. After this, UVR-C comes and it gets bound to the DNA molecule adjacent to the bent angle, and it actually makes two nicks in the DNA molecule in the defected strand. First nick is made around 8 nucleotides upstream and the second nick is made around 4 nucleotides downstream in the defected strand. So actually this UVR-C, it has a dual DNA cleavage activity. After this, after the nicks are made, then the UVR-D protein comes. UVR-D protein is a type of helicase 2. So it can cause the unwinding of the DNA strand as well as it can perform many more functions also. Like it comes and it removes the in-between portion of the defected strand in between the nicks already made by UVR-C. So we have a molecule like this. The correct strand is there, but the defected strand portion has been removed. After this, DNA polymerase 1 comes. After this, DNA polymerase actually attaches the correct nucleotides from 5' to 3' end. And the last nick which is left after attaching the nucleotides here, the nick which is here, this is ligated with the help of DNA ligase enzyme. So this is how the defect gets removed and we have a normal correct DNA molecule here. So this type of repair actually gets done when the transcription occurs. So this is known as the transcription coupled repair, and it actually helps in removing the dimers as well as adducts. See, dimers are formed when the two molecules join, then the resultant molecule is called dimer. When the two similar molecules join, then the resultant molecule is called dimer. Adducts are the resultant molecules formed without the loss of any atom. See, suppose this is one molecule made up of 4 atoms and this is the other molecule made up of say 5 atoms, just to say. When these two molecules join and no atom gets removed from it, and the resultant molecule will now have say 9 atoms like this. These are the atoms of the first molecule and 5 atoms of the second molecule are there. Then the resultant molecule is called adduct. See, there can be rearrangement of the atoms. This atom can shift from here to here, this portion, but the number of atom remains same. So such molecules are called adducts. This nucleotide excision repair helps in the removal of dimers as well as adducts. So this is for today. This is how nucleotide excision repair gets done.